Welcome back from the break. We're going to dive into the rest of the forensic biology and DNA presentations now. So next up is Dr. Jianye G, an associate professor and associate director with the Center for Human Identification at the University of North Texas Health Science Center. Dr. G will discuss the interpretation of Y-chromosomal STRs for missing person cases. Dr. G received his bachelor's and master's degree in computer science from Nanqi University in China. He earned his PhD in bioinformatics from the University of Cincinnati. Prior to joining the University of North Texas Science Center, Dr. G worked in the Human Identification Division of Thermo Fisher Scientific as an algorithm leader and later as a global market development manager. His research relates primarily to computational analysis and interpretation of forensic DNA data. Welcome, Dr. G.
Thanks, NIJ, for giving me this opportunity to share my recent work. This study is about interpreting the Y chromosome STR profiles for missing person cases. And we're going to develop a software to serve this purpose, and we'll share the software for free with the forensic community. So first of all, why do we want to use Y-STR for forensic applications? The first reason is probably because most violent crimes are committed by men. Here's some statistics from DOJ. For example, 99% of the forcible rapes are committed by men. So that's probably the most important reason. And second, Y chromosome is only inherited from father to sons, and it can be used to trace the male lineage. And this unique feature makes Y chromosome very, very useful in many forensic applications like the kinship analysis, missing person identification, familial searching, mixture interpretation, infer the ancestry population, link multiple different cases, and also Y database searching. So in our center, UNTCHI, we have a missing person unit. This unit processes hundreds of human remains samples and more than 1,000 family reference samples every year. As of last summer in Andes, we contribute more than 50% of the remain profiles and about 65% of the reference profiles in Andes. So we process lots of missing person cases. And for those cases, we follow the SWIG-DAM guideline to interpret the Y profiles. So in the SWIG-DAM guideline for missing person cases, this guideline recommends that additional DNA method other than autosomal STR typing, such as the mitochondrial DNA or Y-STR typing, should always be considered if relevant. And it also recommends that a Y-STR-like ratio and a mitochondrial DNA-like ratio are computed for each of those systems. However, the SWIG-DAM interpretation guideline for the Y chromosome STR typing focuses on the haplotype frequency estimation, and it does not give a recommendation on how to calculate the likelihood ratio for complex cases with mutations or with multiple male references. So, but in our lab, because we process lots of missing person cases, it is not uncommon for us to see some cases with mutations with multiple male references. Here's one example. In this example, we have one missing person, D, and two references, B, uncle B, and E is the brother reference, and we don't have the Y haplotype for the father C and grandfather A. In this example, just for demonstration purpose, I just list two markers. The first marker, all these three individuals has 11, but for the second marker, the first reference B has allele 20, and second reference allele is 21. So apparently there's some mutation may be involved in this case. So this is a typical complex case with mutation, with missing data, and with multiple male reference. And we still need to calculate like ratio. And how can we calculate like ratio for such kind of scenario? Many years ago, we in our lab already developed a method to calculate the pedigree-like ratio for lineage markers. But at that time, we didn't have enough resources to really develop software implement the method. Now with the NIJ support, we're able to do that. In some cases, the DNA analysts also would like to would like to quickly determine if two Y profiles are from the same lineage. They don't care if the two profiles are the father-son relationship or first-cousin relationship. They just want to know if they're related or not. We did develop a convenient and accurate method to serve this purpose, and we're also going to implement this method in our software. Next, let's get into the detail of these two methods. Let's see the pedigree-like ratio first. The pedigree-like ratio with Y-STR profiles is the principle is the same as any other forensic evidence. We have two competing hypotheses. We calculate the likelihood of observing evidence given two different hypotheses, and calculate the likelihood, compare with them, then you will get the likelihood ratio. So in missing person cases, we have two hypotheses here. One is kinship hypothesis, one is non-kinship hypothesis. So in the kinship hypothesis, we have missing person. We assume the missing person D belongs to this reference family with specific relationship. In the non-kinship hypothesis, we assume the missing person is unrelated to the reference family. So we have two different hypotheses, two different pedigree, and we want to calculate the pedigree likelihood for each of them. The pedigree likelihood for the Y profile is actually the product of the cumulative haptide frequency of the founders and the cumulative transmission probability of all father-son pairs. But before that, in case there's some untyped individuals in the family, like the example we show here, we have some missing data, right? Untyped individuals, then we need to guess the possible haptypes of those missing person. Like, let's say in this particular example, let's say A could be 1120 or 1121. C could also the same could have the same haptypes, and we need to calculate the likelihood for each haptype combination. So, and there could be there many possible combinations. Let's say if A has 1120 and C has 1121, and we have this one possible haptype combination, then when we calculate the pedigree likelihood, we first look at the founders. In this example, we have two founders, A and D. So the founders haptype frequency would be the product of these two haptype frequencies. And you may get the haptype frequency by searching some Y-STR database like the Weir. For the transmission probability, it really look at the father-son pairs. In this example, we have three pairs, right? A to B, A to C, and C to E. We calculated transmission probability for each pair. And for each pair, each father-son pair, the transmission probability actually is the product of the transmission probability of each marker. Let's say if you have 15 markers, then you multiply the transmission probability of each of these 15 markers. Here we assume the mutation events are independent across all the markers. And in this calculation, we need to know the mutation rate, which can be obtained from Y-STR. And we need to select a appropriate mutation model, for example, two-phase model. And then we can calculate the pedigree likelihood given all the individuals have type known. But this is just one possible haptype combination. If we consider all the all possible haptypes for those untyped individuals, there are probably many possible haptype combinations. Then the pedigree likelihood will be the sum of the likelihood of all those possible haptype combinations. So in this particular example, we have two unknown untyped individuals. Each untyped individual have two possible haptypes. Then in total, we have 4 different scenarios. Then we calculate the pedigree likelihood for each scenario and sum them up, we get the pedigree likelihood. So as you can see, this is not easy calculation that you can do by hand. And that's why we want to develop software to assist our DNA analysts to do that calculation. In the software, we're also going to implement the second method, which is to quickly determine if two Y-STR profiles are from the same lineage. The pedigree likelihood ratio method is good, but it's a heavy solution. You need to know the pedigree structure, you need to know the haptype frequency, lots of information. But those information may not always be available. In some cases, DNA analysts just need to know if two profiles are related or not. They don't care if this father-son relationship or first-cousin relationship. They just want to make some quick decision. So we did study a few years ago, and we provide a simple but accurate solution to serve this purpose. It uses the number of mismatched loci and steps between a pair of profile. And you don't need to define a pedigree structure. You don't need to know the haplotype frequencies. The idea is fairly simple. In general, more distant relationship will have more distant, more mismatched loci or steps. For two Y-STR profile from the same lineage, and usually they have no or very small number of mismatched loci or steps. For example, for a father-son pair, almost 100%, they will have close to 100%. They have zero mismatched steps, or some small percentage of 1 or 2. So very small number of mismatched steps or loci. But for two profiles from different lineage, then usually they have a relatively large number of mismatched loci or steps. Like in this example, this is the mismatch distribution for two unrelated Y profiles. And usually you expect to see 10 or 20 or more mismatched steps between two unrelated pairs. And then we can merge these two distributions together, and these two distributions will come across at some point, and then we can cut the threshold at this crossing point, and then we can make a decision. Say, if you see for a pair of profile, if you see 0, 1, or 2 mismatched steps for a pair of profile, then they are more likely father-son relationship. In this case, I only put the father-son as example. And if you see 3, 4, or more mismatched steps, they are more likely from unrelated. So this is a quick way to help the DNS make some decision. Next, I'm going to use the real data, the Y-file data, to show you how we can draw the distribution and make pick the threshold. So first, we obtain the Y-file profiles from one of our previous publication, and we get the mutation rate from Weir-TriD database, and then we define the lineage as up to meiosis 4, which means the first cousin relationship in this example. And then we draw the with some mathematical model in the simulation, we are able to draw the mismatch distribution for those close relatives. Usually it's kind of going down this way. And also we are able to draw the mismatch distribution for the unrelated pairs, and it's going up this way. Then they are come across at point between 2 and 3. So we can say, okay, for Y-file profile, if we see 0, 1, or 2 mismatched steps for a pair of profile, then they're more likely from the same lineage. But here we define the lineage up to first cousin relationship. And also you can see this is the total population. If you look at African American, Caucasian, Hispanics, or individual population, those distributions are pretty stable. So yeah, this is how we can draw the distribution and decide the threshold. So we provide a quick way to an easy way to the DNA analyst to quickly make some decisions. But the problem is how reliable or how accurate is this method. Here's some data from one of our previous studies. We used more than 7,000 unrelated Y-file plus profiles, and we define a lineage is up to 4 meiosis relationship, and basically first cousin. And we are able to find decide the threshold between 5 and 6. So which means if a pair profile with 0 to 5 mismatched steps, they are more likely from the same lineage. With this threshold, the chance of making mistake is extremely low, because it is unlikely for 2 unrelated Y-file plus profiles will have less than or equal to 5 mismatched steps. And it is also unlikely for all close relatives that will have more than 5 mismatched steps. And in fact, we look at the genotype error in general, it is more likely than the error associated with this method, right? So another thing I want to mention here is that the accuracy will depend on the lineage definition. So here we define the lineage as up to 5 meiosis, but if we increase the number of meiosis, basically define the lineage including more distant relationship like 5 meiosis, 6 meiosis, even 10 meiosis, then definitely we will see the distribution more distant relationship, and they may come across in the more distant, higher number of mismatch steps. So then the accuracy it can be a little bit lower. We're almost done with the interpretation software. We developed all the algorithm, the UI, and we are focusing on the testing and validation of the software. And hopefully the software will be ready probably in end of March or beginning of April sometime. So in this software, we implement the 3 workflows. One is like ratio, pedigree like ratio calculation. Second is the mismatch distribution to help decide the threshold. And also we implement the simple profile comparison, because this is a very common use scenario. That's why we decided to include that. So in the simple profile comparison, you are able to do the one-to-one match, one-to-n and n-to-n, many different options to compare the profiles. And it will show you the comparison results in all the details. In the like ratio calculation, the user is able to draw the pedigree by themselves, and you can assign the once you have the pedigree defined, and you can assign the haptypes to the pedigree, and assign the haptype frequency to those individuals or to the haptypes. And then the software is able to calculate the pedigree like ratio and give the estimate. For the mismatch distribution, you can you can define the meiosis, maximum meiosis, so change the definition of lineage, and you can report by mismatch steps or mismatch loci, and the software will draw the distribution like this. You can zoom in, zoom out to decide where the distribution come across each other, and then decide the threshold. You can also save the picture into a PNG file. So finally, I would like to thank NIJ for funding this project, and thank the support from my colleagues in UNT-CHI. They really help a lot in this project. Thank you so much. Thank you for your time and patience. Thank you.
Thank you, Dr. Yi. Great job. If you have any questions for Dr. Yi, please type them in the chat and we'll get them answered during the Q&A session. Our next presenter is Dr. Julie Valentine, an Associate Dean and Associate Professor at Brigham Young University College of Nursing. Dr. Valentine will discuss an update on the assessment of sexual assault kit evidence selection leading to the development of a sexual assault kit evidence machine learning model. In addition to her work at Brigham Young University, Dr. Valentine is a practicing certified adult and adolescent sexual assault nurse with Wasatch Forensic Nurses. Her clinical specialty and research focus areas are sexual violence, intimate partner violence, and criminal justice system response to sexual assault violence. Dr. Valentine focuses on a multidisciplinary collaborative research studies uniting disciplines in sexual assault case reform to benefit victims in case processing. Thank you for joining us, Dr. Valentine.
Well, greetings. I am eager to present to you some of our findings and our work on our NIJ grant, assessment of sexual assault kit evidence selection, leading to development of a sexual assault kit evidence machine learning model. So I'm going to tell you some of our current findings and a bit also about what we are working on. So our proposed research study is on addressing the gap that we've found in research on evidence selection protocol to establish best practice guidelines for SAK evidence selection for analysis, and today take those findings and test a sexual assault kit machine learning model software program. So I'm sure many of you know that regarding what evidence or swabs are actually tested from a sexual assault kit can vary. We have some crime labs that have a test-all approach. We have some crime labs in areas of the country where the forensic analysts or the evidence collector, such as the sexual assault nurse examiner, will determine what they believe are the most probative swabs. So those swabs that they think, yeah, these are more likely to develop a CODIS eligible profile. That's our outcome variable, both for STS, state database system, and NDIS. And then from there, it might change even more. So a lab may choose to pick 3 of the most probative swabs and test those for quant male DNA or do a Y screen. And then depending on those findings, may move just 1 swab through DNA. So we're evaluating what is the most effective and how do the variables related to patients and assaults impact the development of DNA profiles. So we have 2 main study aims to evaluate the decision-making protocols, somewhat what I talked about and introduced earlier on DNA evidence in sexual assault kits, and then to develop the machine learning model. So our first step is we look at the percentage of SACs that develop a full or partial STR DNA profile and or a CODIS eligible profile. So when I say partial, I mean they have to have enough loci developed that it could go into at least STS or the state database system. We then are looking at the predicting variables for developing a full or partial STR DNA profile. And we are gathering these variables or features from the sexual assault medical forensic examination forms that should accompany crime labs. I have learned that some crime labs do not look at these very much. Other crime labs read quite a bit, especially the what touch what in their decision making about what to test. But we feel that the sexual assault medical forensic exams really contain valuable information in deciding the process of swab selection. And then the next step, and this is what we are fully immersed in right now, is the predicting variables based upon swab location. Then that moves into development of the machine learning model to predict the likelihood of developing a full or partial STR DNA profile with specific swab probabilities. And I'll share an example of that towards the end of this. And we're planning on testing this machine learning model in 3 sites. The data that I'm presenting to you today is all from Utah. So it's the State of Utah Crime Lab, Utah Bureau of Forensic Services, which kudos and hats off to them. They are our partner in this and submitted the grant with us. And then the state of Idaho, love working with them as well. And then Orange County Forensic Lab, and they have been wonderful to work with on this project. So here's our research question and preliminary findings. So our first research question was, well, how many of these sexual assault kits actually develop a full or partial CODIS eligible DNA profile? And you can see this. The findings I'm sharing with you now are cases from 2010 to 2018. We are coding the 2021 because our crime lab now is doing the DNA analysis quickly. And so our N is going to be almost double that, but these are preliminary findings. And some kits never made it to crime lab, even when we asked for all the backlog kits to come in. We know that some kits were destroyed. And you can see that those that were submitted, 44% developed a full or partial CODIS eligible DNA profile. That does not correspond to how many of them actually went into CODIS, either NDIS or STS. So we did a sampling of those cases that did not go into CODIS, but had developed full or partial CODIS-eligible STR DNA profiles. And we found here are a couple of reasons why they did not go into CODIS. One was if a consent sexual partner standard had not been submitted. And I know that there's some subjectivity and variability on crime labs if they will enter a CODIS-eligible profile if they did not have a consent partner. And about a third of our cases, our victims had a consent partner. And then one of the other reasons that we found in several cases was if the foreign contributor DNA profile was developed from a body area that the victim said was consensual. So if the victim said the oral contact on breast was consensual, and that's where the DNA profile was developed, and there was no profile developed from the vaginal swab, and the victim said the vaginal penetration by the penis was not consensual, our crime lab would then not enter that DNA into CODIS. Also, cases where law enforcement then responded back and said no crime was committed. So our next question is, what victim and sexual assault variables are statistically significant in predicting the development of a CODIS-eligible profile? So that's our outcome variable. And we found, first off, that gender is hugely significant. You can see females 38% and males was 19%. And due to this substantial difference between male and female, we actually have divided our research now into male cases and female cases. And if you wonder why this is, I mean, just think of anatomy. Males don't have vaginas, and vaginas will hold on to more of that DNA, we believe, than a male, either penis or rectum. So we are finding quite a few of the male cases we are not developing CODIS-eligible profiles. The next we looked at was age. We found a substantial difference with as women age, and you can see this chart here. And that bottom number, age of 10, we actually start at 14 with our data set. So between the ages of 14 to 30, where women's estrogen levels are staying relatively about the same, we have the level of developing a CODIS-eligible profile, meaning from the foreign contributor, is about the same. But as a woman ages, there's a decrease in estrogen, especially when a woman goes through menopause. That decrease in estrogen causes a change to genital tissues. There is not as much secretions or lubrication produced. The tissues change. And we believe that those changes that happen also mean that they are less likely to hold on to foreign contributor DNA. We also wanted to look at bathing or showering. 37% of the females bathed and before the exam, after the assault, and before the exam, so a pretty high percentage. And I'm a forensic nurse, so I'm still taking care of patients. And many times they will say that they just wanted to wash off what had happened to them. And they really don't think about, oftentimes, am I going to report for sure? Am I not going to report? So a high percentage do bathe. So you can see that bathing or showering certainly impacts the development of a CODIS-eligible profile, in fact, by half. If no to bathing and showering, 42% developed a CODIS-eligible profile. We then looked at bathing and showering in relationship to time between assault and the exam, thinking that the longer it went, it's more likely they will have bathed and showered. And that's exactly what we found. There's a strong correlation between bathing and showering and time between the assault and exam. We also looked at if they had bathed or showered, what was the likelihood of external swabs producing CODIS-eligible profile, and then internal swabs. And it makes sense that we're more likely to see 38% from internal swabs compared to 25% of external swabs. But I want to clarify, that still means that it's 25%. So we still collect. We have a case where a woman had showered. She had her examination 3 days after the rape, 76 hours actually after the rape. And she had told the SANE that she had been kissed on the neck. The SANE swabbed the neck, and they found a full STR DNA profile more than 3 days. This was from oral contact, more than 3 days after the rape, after the victim had showered. This victim also had a STR profile that had been developed from the vaginal vault as well. So then we wanted to look at, in the sexual assault medical forensic exams, we've got a whole bunch of data. It's not just what touched what, but a lot of information that we collect from victims for their health care, because that's the number one reason we do these exams, but also related to what happened to them and to guide our evidence collection. So I'm going to share with you statistically significant variables that we have found from these charts. Strangulation. Strangulation, if the victim reports being strangled, we are more likely to develop a CODIS-eligible profile. This does not mean just from the neck, just this means overall strangled. Grabbed or held. Both strangulation and grabbed or held means the victim had more epithelial DNA contact, possibly being grabbed and held, as well as with the strangulation. Higher number of assaultive or penetrative acts. So we count up to 4 penetrative acts or assaultive acts. That is for a female, that's going to be penetration of vagina or rectum or oral mouth, or if the perpetrator had oral contact to the female's genitalia. The more penetrative acts, the more likely we are to develop a CODIS-eligible profile. Ejaculation. Now, first I want to talk about this unknown of 53%. When I do trainings with law enforcement, I share this number. I say, more than half of our victims, when we ask them, did he ejaculate? They will say, I don't know. That's a sign of a non-consensual act, because most people in consensual sex know if their partner orgasmed or ejaculated. So unknown in 53%. Now, if they say yes, then 47% develop a CODIS-eligible profile, compared to if they say no, 26%. A non-anogenital injury. About 75% of our patients have non-anogenital injury. We found that that's associated with developing a CODIS-eligible profile. If they state that there was penile, vaginal penetration, and oral contact by suspect on the victim's genitals or breasts. We then looked at those variables that are less likely, statistically significant, to develop a CODIS-eligible profile. All of these are somewhat related, meaning if a victim reports loss of consciousness or awareness, we actually have 49% of our victims that report loss of consciousness or awareness, everything from being strangled to drug facilitated to intoxication by drugs or alcohol, or we have many cases where victims say loss of consciousness or awareness, actually 25% of our cases where they didn't have alcohol or drugs or suspected drug facilitated or were strangled. So we think this is actually from the trauma of the assault. But if they have these gaps in memory, they are not able to tell us the what touched what. And so we have found that they are less likely to develop a CODIS-eligible profile. So if a victim says unknown, meaning that they had a full loss of consciousness and they don't know who sexually assaulted them, less likely to develop a CODIS-eligible profile. Same with suspected drug facilitated. Condom use. Again, condom use, only 10% of cases. In the American College Health Association survey, they found that 47% of college students use condoms in consensual sex. Again, this low percentage of using condoms, I think, equates to non-consensual acts. But you can see if they use a condom, they are less likely to develop a CODIS eligible profile. And then multiple suspects, more likely to develop a mixture and more difficult, even though this is improving, to tease out a CODIS eligible profile. So we've also looked at crime lab statistically significant variables. We have found so far that the more items or swabs are tested, the more likelihood that the kit will develop a CODIS eligible profile. We looked at the STR DNA kit used, and you can see we found a substantial difference between ID Plus and Global Filer, but ID Plus was being used when our crime lab was doing serology first, and so those kits that were moving on had a positive serology, rather going direct to DNA. So additional findings of interest, we push to have sexual assault kits submitted and tested for justice, and we frequently talk about justice for victims. We have 84 sexual assault kits where we had a suspect standard, and they developed STR DNA profiles that excluded the suspect. There was less evidence against that suspect to charge them in the crime. So we test sexual assault kits for justice. It's for justice for victims, but overall justice for perpetrators too, that we can really look at what is the DNA. It's using science to improve justice. So the next steps on data collection is we are completing data entry. We have about 1,700 cases from the Idaho State Police Crime Lab. We are almost done coding those. We have gotten about 1,500 cases from the Orange County Crime Lab and forensic nurse specialist. So huge thanks to both of those partners. And we are coding that data. And then we will be looking at the same things that we have looked at here in Utah with these other data sets from Idaho and Orange County. Are we finding the same predicting variables? We're then working with our state crime lab to do 6 months as a pilot study of testing everything, all swabs. And we are working out the process and decision-making steps for that. So our next steps is, as I stated before, to evaluate the swabs, to develop the machine learning model, and then to test them in the 3 sites. This is just an example of how we see this machine learning model providing feedback to crime labs to help with throughput and to have guidance on what to test on sexual assault kits if there's a selected number of swabs tested. So you can see in this case, vaginal vault was 77% probability of developing a CODIS eligible profile, and it goes down to what the oral swab would be. So if this is effective, a crime lab could utilize this machine learning model, which if it's effective, we will make this open to anyone that would like to obtain the model, this machine learning model. And it could help to guide if they are selecting their number of swabs and reduce the time by forensics analysts in reading the what touched what and the crime and going through the case and then determining which swabs to test. So our other research question, what differences exist between the forensic scientists in the selection and prioritization of swabs for analysis? We are developing a survey with case studies, which we're hoping that a number of forensic labs will be interested in utilizing. And then we'll evaluate inter-rater reliability. And here is my contact information. I look forward to meeting with you later during the question and answer session. Thank you.
Great job. Thank you, Dr. Valentine, for your presentation. Our last forensic biology and DNA presenter is Dr. Bruce Weir, a professor of biostatistics at the University of Washington. Dr. Weir's presentation will center on the interpretation of Y-STR evidence. Dr. Weir is a fellow of the Royal Society of London. He is the co-author of Interpreting DNA Evidence, an early text addressing DNA evidence. He has also published over 200 peer-reviewed papers in statistical genetics, with many of these papers focusing on forensic genetics specifically. Dr. Weir chairs the DNA subcommittee of the New York State Commission on Forensic Science, and he also directs the Genetic Analysis Center at the University of Washington, which acts as the data coordinating center for a large genetics consortium funded by the National Institutes of Health. Welcome, Dr. Weir.
So hello, I'm Bruce Weir, and going to tell you about some work we're doing on the Y-STR evidence and its interpretation. This is work with my colleague John Buckleton in New Zealand, and a lot of the work's been done by some students working with us, and those students and John and myself are supported by this NIJ award. So we have some principles to help us set up some methods for interpreting Y-STR data. The first one is that we're going to approach evidence by looking at the probabilities of the evidence under alternative hypotheses about the contributors. For example, for a single contributor stain, one hypothesis is that the person of interest was the contributor, and the alternative would be that no, someone else was the contributor. It does help, though, to articulate those hypotheses. And then we calculate the probability of the evidence under each hypothesis. And we have to keep in mind that if the hypothesis includes an unknown or untyped contributor, then the probability in fact depends on the profiles we've already seen, on the fact that someone else has been seen to have that profile. We do want to take account of the whole evidential profile, all the loci that were typed we think should be used in our interpretation. And we also would like to keep in mind that these Y-STR profiles are genetic entities. Their frequencies are shaped over time by things like genetic drift and mutation. So by far the simplest approach and the most common one is the so-called counting method. So it's a crime, there's a Y-STR profile, and that's queried against the database of profiles. And currently in the US, we make use of the European database, the YHRD. It's online, it's large. At the moment, there's 283,000 profiles for the Y17 data set using the PowerPlex Y23 kit. So anybody can go to that website and upload or manually enter a profile and get a report. The US forensic scientists mainly use only the US data within the European YHRD dataset. And the corresponding US data has 29,000 profiles for the Y17 dataset and Y23 PowerPlex kit. So here's a little example. This is an actual PowerPlex 23 profile, but it was only partial. So there was results available only for 19 loci. And that was entered into YHRD, and no matches were found. In fact, no matches were found in the whole data set, but in particular in the US data, there were no matches. So we see statements like no match in 7,000 haplotypes for African American. So that 1 in 7,000 would be an estimate of the population frequency of that type. And then there's an upper confidence interval. So the fact that it wasn't seen doesn't mean that the profile doesn't exist, of course, in the population. And the upper confidence interval is 1 in 2,000. So the population frequency could be as high as 1 in 2,000, and we still not see it in the sample of size 7,000. So there are results given for, looks like 5 major ethnic groups, and then a single number using all the data. So that's a useful summary of the data. It's useful, but it doesn't really address any of our guidelines. So it is an estimate. It's a good estimate of the probability a random man has that particular profile. But it doesn't take into account that we already know that the profile is carried by at least one person, typically the defendant in a trial, the suspect before we get to a trial. It doesn't take explicit account of the hypothesis about the source of the profile. It may not use all the loci that have been typed. So we have a lot of discussion about reduced locus matches. So it doesn't use all the information we have, and it doesn't really make any take advantage of any of our information about genetic profiles. So the counting method is very dependent on the size of the database. If we change the database size, we add a few more profiles, we'll change the answer, not only the potential number of matches, but certainly will change the confidence limit. More importantly, though, we get the same answer for different numbers of loci. So for that one example that we showed, the 19 locus profile, we reduced the profile by taking out one allele at a time, and we got all the way down up to and including 8 loci, and we still found no matches in the database. So whether there was 8 or 19 loci, the counting method gives the same answer. And that seems to be a pity, because we would certainly believe, I think, that the 19 locus match is much stronger than an 8 locus match. So that's certainly a very common method. It's valid, but I don't think it really addresses the forensic issue. A method that is closer to the forensic question, giving a match probability, was given by Charles Brenner, and Y-HID gives results for using Brenner's kappa rather. So instead of taking 1 over the database size, the match probability is reduced by this quantity kappa, which is the proportion of haplotypes in the database which occur once only. This is really designed for the common situation where the evidential profile has not been seen in the database. So it's added to the database, and in this augmented database, what proportion of the profiles occur once only. So if this was if kappa was 0.5, then the match probability would be estimated as 1 over 2n, as opposed to the 1 over n for the counting method. It's a valid method, but it's a little restricted. It doesn't extend as easily for situations like mixtures or making use of relatives. And once again, it depends very much on the size of the database. We did the same thing with that previous 19 locus profile. This is what we find using the US data. We now find the kappa estimate is 1 in 16,000 for the African American. So that's certainly it's less than the 1 in 7,000 from the counting method. However, when we reduce the number of loci from 19 all the way down to 8, we still get the same answer. So the kappa method gives us the same answer, regardless of how many loci were typed. It depends only on the number of singletons in the database. So what are the issues? And I think the main issue is this distinction between profile and match probability. So here's a couple of hypotheses that we might have about a single contributor. H1, the person of interest, the suspect, the defendant, was the contributor to the stain, versus the alternative, some other man was the contributor. And then the likelihood ratio compares the probability of the evidence under each of those two hypotheses, and it reduces down to the reciprocal of this bottom line, the probability that an unknown or untyped person has the profile of interest, given that we've already seen someone with that profile. So AA is the haplotype, and this is the conditional probability. So this is the likelihood ratio, whereas the counting method and the discrete Laplace method which we won't go into here, they really address the profile probability, not the match probability. So it's just they are answering different questions. This seems to be the relevant forensic question. So how can we take advantage of information we know about population genetics? So these Y-STR loci, when they're typed by capillary electrophoresis, keep track of the number of repeat units at each locus that are detected. And the number of repeat units can change by mutation, and they typically change by +or -1, or maybe +or -2. So this is called the stepwise mutation model. And it was the same theory that was developed and discussed back in the 1970s for protein variants. At that time, electrophoresis was still used, but it was on a gel, and the molecules moved it across the gel according to their charge, not their size. So a single amino acid change could cause the net charge on the protein sequence to change by +or -1, or maybe +or -2. So there was a model developed at that time for allozyme data, and Moran showed that the distribution of allele frequencies under that model wandered. So the shape of the distribution was sort of fixed, but it wandered up and down. We couldn't predict what the mean or the mode would be. The variance had a predictable value, but not the mean. And that sort of argues against the logic behind the discrete Laplace method, which looks to estimate the haplotype frequencies. The matches, however, it's the match probability we really need, and that really that's like a moment, like a variance of the distribution. And Oda and Kimura gave a nice little formula for the predicted value of the probability of two alleles in that case, taken randomly from a population, matching, being the same. They call it homozygosity. Use the mutation rate, and they looked at single-step mutations, so equal rates μ/2 in the plus or minus direction. Population was undergoing random mating, so it's an equilibrium or a balance between loss of variation by drift, increase of variation by mutation. So the prediction from this theory is that the logarithm of the chance of two Y-STR haplotypes matching is a linear function of the mutation rate. And it goes down. So the more mutation there is, the less is the chance that two haplotypes will match. So, of course, mutation increases with the number of loci. So, in other words, the more loci there are, we can predict that there'll be less matching. This matching proportion, or what Oda and Kimura called homozygosity, is essentially the quantity that we call theta in the forensic literature. This is often called the population structure parameter, but it's really the probability that two, in this case, two haplotypes are the same because of their descent from a common ancestor. The match probability is bigger than the profile probability. So here's the match probability that we are interested in. Here's the profile probability that the counting method produces. And the relationship between them is a very simple one, depending on theta. The match probability is bigger than the profile probability. So the likelihood ratio will be smaller using the match than it would be simply by using the counting method profile probability. So once we've got a model in mind, drift and mutation, it's called the Wright-Fisher model. And it's the model that Anderson and colleagues used in the discrete Laplace method. Once we've got that formulated, it's very easy to simulate data. So here's some results from a very simple simulation. We had a population of size 10,000 haplotypes, 20,000 people, I guess. 20,000 people. So, and the mutation rate of 10⁻², fairly high. And 50, we looked at 50 Y-STR loci, no recombination, of course. We let this run for 100,000 generations. And we plotted the matching proportion. So we looked at all the possible pairs of haplotypes, 50,000, 10,000, taking 2 of those, so that's about 50 million comparisons. What proportion of those pairs of haplotypes match at exactly 10 loci, or 20 or 30? It's interesting that the lowest proportion is actually for 0 matching. The proportion of pairs of haplotypes that match at one locus is higher than it is for 0, and then it starts going down. And after 10 or so, we can't really distinguish graphically. They're quite small. It's better to take the log, it's just minus log 10 of these numbers. Here they are plotted, still against the number of loci. And so the theory says that this log 10 matching proportion should be a simple linear function, should be a straight line with the number of loci. Well, it's not. It starts out that way, and then it sort of bends over. So we sort of get less benefit by typing more loci as we do that. We continue to get a smaller match probability, so the evidence becomes stronger, but it does so at a diminishing rate. So that's interesting. Not really surprising, I think, but we'll come back to this point. Here's something which is a more recent way of thinking about it, and it sort of follows on from the previous slide. If we get 2 haplotypes that match, say 20 Y-STR loci, why is that? Either they're from the same man, or if not, they're from the same male lineage, so they're from related people. And once we have that possibility, then we would sort of expect to get more and more matching. So the more matching we see, the more loci that match, the more loci we expect to see. So what we've done here, with the same set of simulated data, we looked at all the haplotypes that matched at, say, 10 loci, and asked what fraction of them also match at 11 loci. So they match at 1 more. So this is this value here, and it's about 60%. So if you've got 2 haplotypes that match at 10 loci, this is a 60% chance they'll match at 11. And this increases. It's a bit noisy. We need to do more simulations, but it's going up. It gets way over 90%. By the time we get out to 40 loci or so, we've got a very high chance of continuing to find matches. So this gives some comfort in using partial profiles. If we get a high degree of matching already, we expect to get more. And then going out, instead of asking what's the chance that they match at 1 more locus, we say what's the chance they match at 5 more. So they match at 20, what's the chance they match at 25? It's just a bit over 50%. So that shows that match probabilities are not independent among loci. We continue to get a benefit by typing more, but the benefit rate decreases. So we have some theory. The theory matches data, as we'll see, and it lets us do a lot of playing around with different methods. Never as good as real data, of course. And we've got this wonderful resource, the YHRD database, 233,000 profiles, but we can't use it. We can type in upload a single profile, but we can't download the data, and that's because of European privacy laws. So a tremendously good resource, and it's not really available for us to do numerical experiments. So we are constructing our own database. YHRD has a list of publications. We're working our way, we've just about finished working our way through those, looked at all those publications, and identified the ones that had the actual profiles published, generally as supplemental material. And we've done over 150 papers so far. About 100 of those have data that are already in YHRD, and we're over 50,000 profiles so far, so we can do 1.25 billion pairs of profiles. And we're continuing. We won't have much more, but we might get up to 70,000 profiles. So we're cleaning the data. lot of mistakes we find, but made by the authors, we think, a lot of uncertainty in what they actually mean. But what we will be able to do is estimate the parameter theta for up to 22 loci for worldwide populations. This is repeating work we did earlier for the autosomal profiles. So that work is progressing well. And here's just a flavor of what we've been able to do so far. We've divided the data, and we looked at it twice, once for 70 profiles that are all typed to 17 Y-STRs, or those typed the full 23. So we did the same thing, look at all the possible pairs of profiles for 3 groups of people, 7,000 African, 21,000 European, and 7,000 South Asian. Each of those 3 data sets, we did all the pairs of comparisons and see what fraction of the pairs match at 5, 10, 15 loci. And then did the minus log 10. So we start out, we get less match. This is on the log scale, so sort of have to think backwards. We increase our matching up to 5 loci, and then after that we start to decrease. The interesting thing is it doesn't make much difference whether the profiles are from Africa, European, or Asian in this case. Now we have the data, and we'll be able to do the interesting comparisons for the smaller populations, particularly those from indigenous groups. So we are building up a good understanding of Y-STR profile matching on a worldwide scale. So that's what we're doing, combination of genetic theory, empirical data, addressing Y-STR profile matching from a sort of a standard forensic viewpoint, constructing likelihood ratios, making use of match probabilities. So thank you for listening.
Welcome back, everybody. And we're about to start our final Q&A session for this section. Thank you, Bruce, for that. Appreciate it. And so Dr. Valentine and Dr. Guy have joined us live and here on camera, and Bruce is able to join us by phone. So I'm certain everybody will be able to recognize Bruce when he answers those questions. All right, so we're going to start, Jianye, with you. Can you tell us about some of the limitations to calculating pedigree likelihood ratios?
So thanks for the question, JC. So the pedigree likelihood ratio approach is kind of a heavy solution. We need to know lots of information, like we need to clearly define the pedigree, who is related to who. And we need to know the haplotype frequency of all the available haplotypes, but which may not be available in many cases. And for some particular population, you may not have enough data to calculate haplotype frequency, right? That's one limitation. And another thing is that when the pedigree is getting big, and not everyone in the pedigree may get typed, so you have some missing data, untyped individual in the pedigree. When those number of the missing data is getting missing individuals, the individual with missing data is getting big, then the algorithm is getting very complicated. It may take very long time to calculate. That's another limitation of the pedigree likelihood ratio. So basically, the pedigree structure clearly defined, which may not always available. Haplotype frequency may not be always available when the pedigree is getting big, and the computation may take a long time. So.
Okay, thank you for that. Julie, I'd like to ask a question from the audience. So I know when you're presenting the information, you were speaking about the fact that you have sort of a limited pool of data for this initial presentation, but you'll be adding additional data that's being gathered. And so the question is, are there, I guess, anticipations that you'd be able to sort of extend the range for those pediatric cases, because you don't have very many data points for those types of cases right now. Just wondering what your thoughts are on that.
We actually are looking at doing that, and that will be with some Utah data, possibly from other pediatric hospitals and Orange County. Our IRB is, and our memorandums of understanding are with forensic nursing teams that conduct exams on 14 years and up. And that's why we are at that 14 years. The national protocols say adult and adolescent patients are kind of lumped together starting 14 years. But there's this huge research gap, right, on the DNA findings and the predicting variables for these pediatric cases. And so we actually have begun our collaboration with Primary Children's Hospital here in Utah, and specifically our first area that we're looking at is the 0 to 5-year-old cases. And we are pulling those and applying for IRB addendums and approval to launch on that study.
Yeah, if there's a group that we need sort of to be able to model and understand how to be the most successful, I think it would be that age group, right?
Right, absolutely.
Yeah. Bruce, a question for you, if you're able to. So likelihood ratios address the probability of a match profile. Can you tell us why, or reiterate why it's preferred in forensics to use a profile probability? So Bruce, we're not hearing you. You may be on mute. There you go.
Yeah, I'm not quite sure. Just could you restate that question? So why do we use a profile?
So you talked about sort of the probability of a matching profile,
right.
But then there is a preference to use the profile probability. Can you kind of reiterate what the difference of that is?
Oh, thank you. Yeah, so I think it was a lot easier at the beginning when we started this DNA typing to think about the profile. What's the chance a random person has the profile? That's a very natural thing to do. And it's not wrong. It's not really the forensic question, because we have someone that we know matches. So, just so before we start, all these profiles are very rare. So it sounds very surprising when we get a match. But when we get a match, it means we've seen the profile once and then we see it again. So that's a very different question. And I think it's really the relevant one.
Got it. Okay, thank you for that. Appreciate it.
Thank you. Thank you.
So, Julie, I have a few more questions for you from the audience, specifically about the reduction in CODIS eligible profiles with increasing age. You had mentioned specifically some of the changes in the vaginal cavity and the vaginal vault, specifically. And so the question is, do you think that it's due to hormonal factors or the number of alleged assailants that you're seeing those particular
or the number of alleged assaults? So we do have,
yes.
So we do decrease in our number. Our prime age, about 55% of our patients are between the ages of 17 and 25. So definitely younger women are more vulnerable for sexual assault. So we have less cases, but we've been doing some preliminary looking with our increasing case numbers, and we are still finding this substantial change related to age. We think that it is multifactorial, though. It likely is not just related to the hormones. When we go back and we look at those examination forms, we find that particularly over the age of 65, patients have a lot more, when we ask what happened in the assault and the what touched what, they have many more unknown answers. I know I did an exam on a woman who was over the age of 85. She did not have dementia. When I asked her what had happened, I think partially due to age, she had a very difficult time finding the words to tell me what happened. And her words were, he did something very uncouth to me, and really was not able to verbalize much more than that about what actually happened. And so we know in cases where victims have loss of consciousness or more unknown answers, we then don't have the direction on where to collect as well, or really what happened as much. And so we see a decreased development of CODIS eligible profiles in those cases. So we think it's a combination of this hormonal change, which I think is physiologically a significant impact, as well as cognitive changes in being able to feel comfortable answering the questions. That makes sense?
Mm-hmm. Yeah, speaking of that, I personally sort of had this question, and you know, you spoke to some of the trauma as you're doing the exam, and as I'm beginning to understand sort of these trauma-informed examinations, the victim may not be able to express exactly what happened during that examination. And so if a period of time goes by, they may recall more details or have more information to provide, which can be impactful. And so they're able to provide that typically through a victim advocate or some other kind of information, so that, you know, as their case is developing, they have that information. But how can you, as a researcher, then account for maybe some of those additional informations that are provided by victims as they have time to reflect and recall on what's happened?
Right. So one of our other studies is we are looking at how many victims report loss of consciousness or awareness and the predicting factors for that. We actually find almost half of the patients state yes to at some point, did you lose consciousness or awareness? We've looked at predictors. We know that if they've had alcohol, if they've been strangled, if it's suspected drug facilitated, those are predictors. But that model does not predict how many patients say loss of consciousness or awareness. We think it's about a quarter that it's from the trauma. Personally, as a nurse, what I do when a patient's really struggling to remember, sometimes those traumatic memories are linked to senses. So I'll sometimes say, what did you hear? And, you know, they might say, oh, I remember hearing the door lock. He locked the door. Or what were your eyes looking at? So sometimes when we are trying to help a patient, we never want to feed anything to a patient at all, but when we are directing our exam, I might ask some of those questions, which have been helpful.
Mm-hmm. Okay, thank you for that. I appreciate it. So Dr. Key, I have one question for you. As you're developing the software, and it's great that you're going to make it available for folks, you know, to use and utilize, how are you imagining those profiles getting into the software? And will they be retained if you wanted to do, say, like, you know, as you're working through a case, you have some information, but you gain additional information at a later date and time, and you want to re-examine or redo the analysis? What does that look like?
So we want to keep this software as simple as possible. We do not want to make a case management system. So what we do is we allow the user to open the profiles through a CMF file or Excel file. You just click open the file, and there's few workflow, as I mentioned in the slides. You can pick one of the workflow to work with, and it will give you the results, and you can export the analyze the results to a file, so we can keep the file as your results, put somewhere you would like to. But our software will never keep any of those data. We just open the file, do an analysis as you want, and export the results. That's it.
Okay, thank you for that.
Mm-hmm.
So Bruce, I have another question for you as we're finishing up our Q&A here. So do you have a goal in mind for the number of profiles that you'd like to have in the database that you're creating? I mean, you had mentioned specifically Y-STR, but this sort of new database that you're developing, do you have a goal in mind?
I had this naive thought we might get 100,000. I think we're probably going to get 70 or 75,000. And that will give us a lot, you know, what we're doing is doing the comparisons. So even 50,000 gives us, you have to multiply those two numbers together. You get hundreds of millions of comparisons. I wanted to get up, you know, over 1 billion comparisons, so that we could talk about probabilities of 1 in a billion. So I think we're going to get very close to our goal.
Fantastic. I know haplotypes can be challenging. They're not quite like STRs, right?
Right.
Okay, so Julie, I have another question for you. So this is another one from the audience, specifically with the 25% of external swabs that give a CODIS eligible profile after showering. Did that include external genitalia swabs?
It did. We put together all the external swabs. When we look at body swabs, the main sites that we see to develop CODIS eligible profiles, number one, breasts, and after that it's neck. And so we did the external genitalia and the breast and neck, lumped all of those. Internal, we looked at being rectum, vaginal. So that's how we divided that up.
Okay, thank you. We have a few more questions, and they're all for Julie right now. I mean, obviously, sexual assault kits are a challenge that forensic laboratories have been working for years to solve, right? The efficiency, all that. And I think this machine learning model is going to be impactful in helping laboratories make decisions. So specifically, there's a question from the audience about sexual assault samples being analyzed with a direct-to-DNA approach. You know, obviously the laboratory protocols are changing as you're working on your research. And so you had mentioned specifically the change from identifiler to global filer, the use of serology, and when they moved from identifiler plus to global filer, did they then go to just a direct-to-DNA approach across the board?
Yes. So the crime labs, all the crime labs that I'm working with are direct to DNA. I know that there are some areas of the country, and in Utah, we do look at pediatric cases differently oftentimes. So there might be serology done on peds cases, but the cases that I'm looking at currently, the 14 years on up, are direct to DNA. I think that will be changing as we look at this 0 to 5-year-old.
Yeah, no, understood. Obviously, having some additional context would be helpful.
Right.
And so it looks like we've got another question that came in for Bruce specifically. We've got maybe one more minute and we'll ask Bruce that question. And while we're doing that, I'll just remind everybody there are some lovely posters. So please go check those out if you have time during your lunch break. So Bruce, regarding match probabilities and accounts for seeing a profile given that is already seen, is that referring to seeing the evidence profile and the suspect profile, or the Y-STRs is seen multiple times down a paternal line? So.
No, I was thinking about seeing potentially two people, the person of interest, the suspect generally, and then if that man is not the perpetrator, then the true perpetrator. So it's two, you know, remember, we always do calculations from the defense perspective. So we're talking about two different men having the same profile. Now, they may indeed be in the same lineage, but generally we don't know that.
Yeah, no, understood. Okay, appreciate that. All right, and with that, our Q&A has wrapped up, and so I hope everybody enjoyed the session. This afternoon, Daniel McLeod-Henning will be presenting in the afternoon session. And so enjoy your lunch, everybody. And